Hi, everyone. Welcome to today's webinar. I'm Dr. Maggie Lynn Bartlett. I'm thrilled to be your host for the Global Virus Network's Forefront in Virology webinar series. We have a really fantastic session today featuring Dr. Domingo. Before we begin, I just want to thank the sponsors of the GVN who's made events like this possible and acknowledge the attendees that we have tuning in from all over the world. Your participation is really what makes these webinars a success, and we're really grateful to have you today. Just so you know, this session is being recorded and will be available for viewing after the event. If you have any questions during the presentation, please type them into the Q&A box, and we will address as many as possible during the Q&A segment. And now I'd like to introduce our fantastic speaker, Dr. Esteban Domingo, who is an emeritus professor at the Spanish Research Council in the Center for Molecular Biology in Madrid, Spain. And during his postdoctoral work, he worked on the genetics of a lot of fun bacteriophages, which led to the first calculation of mutation rates for RNA viruses, and was really the first experimental evidence of quasispecies dynamics, which is something that is absolutely fascinating to me. His documented work not only indicated high error rates in quasispecies of animal and RNA viruses, but really opened the way to how we understand lethal mutagenesis in kind of an antiviral strategy. He is very well published of numerous research papers and books, and has been honored through a number of organizations, from serving on the National Academy of Sciences, vice president of the Royal Academy of Sciences in Spain. And we're just so grateful that we get to hear from Dr. Domingo today and have him enlighten us on quasispecies. So without further ado, I'm going to hand it off to you.
Thank you very much, Maggie. Thank you. Thank you to GVN for the invitation. I'm very happy to be here and to talk about our work of several groups that we work as a consortium. If somebody has questions during the talk, please let me know, make some sign. I only see my screen now, so I don't have contact with the outside world, but you can, tell Maggie and interrupt. Also, you will see in this first slide, there is my email address. I'll be happy to respond to further questions in the coming days, or if you want to have some literature, paper, whatever. So our work, as Maggie has said, is dealing with the concept of quasispecies in RNA viruses and of error catastrophe, which is a theoretical concept that little by little is becoming more and more a part of the antiviral strategies that we use to combat viruses, okay? And because Robbie told me to really give some catchy title, I have put be or not to be in the title. And after I saw that the title was accepted, I thought, gee, I have to justify that. And I'll do that justification just at the very end of my presentation, because it will be a kind of recap of what I have said. So the plan of my talk is the following. I'll talk about what are viral quasispecies and why are they relevant to medical virology. Second, complexity of SARS-CoV-2 isolates and incipient functional diversification. Third, clade discordant residues in SARS coronavirus 2 populations, mutant spectra as information repositories, which is some interesting new thing that is coming up with the millions of sequences that we have of SARS coronavirus 2. Then I will talk about error catastrophe and synergistic lethal mutagenesis. That's work we are doing at the moment. And finally, I'll finish with some conclusions. So viral quasispecies. Viral quasispecies means the ensemble of genomes that are mutated and that replicate inside each infected organism, be a plant, an animal, human, when it is infected by a virus that has low fidelity replication. And viruses with low fidelity replication are, until now at least, all the RNA viruses that we know, that are obviously the cause of very important diseases. So as shown in this cartoon, even if you infect an organism with a virus with a defined sequence, rapidly it becomes an ensemble of compartmentalized genome distributions that we called a mutant spectrum or different mutant spectra within an individual, and they are shown schematically on the right. Each line represents a genome, and each colored symbol or line represents some type of mutation, a point mutation, a deletion, transition, transversions, what you want to imagine. But you have to imagine now that instead of these about 20, 30 lines that I have drawn here, each infected organism can have 10 to the 10, 10 to the 11, 10 to the 12 genomes of this sort during replication. So it's really, difficult to imagine that complexity and that ensemble of mutants that is the reality of RNA viruses is actually the extremely complex collection of genomes that has a number of biological consequences. And in our laboratories, me and other colleagues I'll mention along the talk, we are interested in understanding the extent of such heterogeneity, its dynamics, and its consequences for viral disease. This is essentially, the objective of our work, to understand this, why it happens, how it happens, and the consequences it has. I want to say that the basic parameter, not the only one, but the basic parameter is the mutation rate. As indicated in the slides, about 10 to the minus 4 mutations introduced per nucleotide copied on average, right? But the most fascinating thing that captured myself many years ago was that if you look at the consensus or the average sequence of that distribution is exactly the same that you had at the beginning in the infecting virus, okay? So it's as if nothing had happened, yet the reality is that you have this huge collection of mutant genomes. But of course, as indicated in the slide, viruses, RNA viruses included, vary not only by mutation, but also through recombination and reassortment in segmented genomes. For example, in the case of influenza virus that you can recognize on the left of your screens with the 8 segments blue, 8 segments red, and the reassortments that are one of the reasons why we have periodic new pandemics of influenza virus, and then recombination, which is very important in some viruses, including SARS-CoV-2, as indicated on the left, top left, where you have a mosaic genome that essentially is due to the jumping of the polymerase from one template to another. So that's the panorama of molecular mechanisms of virus variation. A bit more detail in a recent review that is now just in press. A few days ago, it was accepted for publication. We emphasize that if we have, for example, a genome of 10,000 nucleotides and an average of 5 mutations per genome, there is only a very small percentage, about 0.67%, of genomes that will have no mutations. So when you look at the whole genome, to have a mutation is the norm. The rare is the genome that does not have a mutation. But if you keep that average number of mutations the same, but you look at shorter pieces of the same RNA virus, little by little, you find that the unmutated or non-mutated sequence is truly representative. So in that left panel, it is a kind of a Poisson distribution of the probability that a genome has 0 mutations or 1, 2, 3, given here by the number K. And you can see that when you look at the whole genome, you have very few genomes without mutations, but if you have shorter stretches, 3,330, then the majority of regions that you analyze are mutated, which means that, for example, if the 30 nucleotide is an epitope for that particular isolate, that epitope will be faithfully represented in all the distributions. This in all this, the mutants from the distribution. Of course, this is with the assumption that we have a random distribution of mutations, but the reality, as you obviously know, is that there may be fitness effects of mutations. There are mutations that may increase replication or mutations that may decrease replication. And of course, if we could consider fitness, we would have a distortion of this type of, distributions of genomes. Of course, the question is now, okay, we look in detail at this, but why is this relevant? Okay, there are several reasons why this is relevant, let's say, to the medical virology, okay? And a part of my talk at the beginning will be to justify this. The first thing I want to emphasize is the problem of sampling. When you have an animal, in this case, swine, for example, infected, you have a virus, huge collection of genomes, but unavoidably, when we study the virus, we have a virus sample that is a subset of the entire population that you have infected organisms. It's obvious. We always work with some uncertainty. With that virus sample, we can do things. We can do what we call experimental evolution, and that's how we learn a lot of things. We can apply selective pressures, antibodies, drugs, changing cell types, changing temperature. We could do plaque-to-plaque transfers to have a kind of sequential bottlenecks that result in decrease of infectivity, and so forth. We have learned a lot of this, a lot about these approaches, but of course we have the limitation of sampling. Unavoidably, our research is imperfect, not only for this reason, but also for these reasons. And another point that we have been reflecting a lot in our laboratory is the relevance to emergent infections. And in the paper that is quoted here in RNA in Press, we make the following distinction. It is very difficult to prove that quasispecies is important for the beginning of an emergence infection. In other words, an emergent infection has three steps: establishment, and dissemination, or if you want, the first contact, and then establishment and dissemination. So to prove that indeed a quasispecies is relevant to the beginning of an emergence is extremely difficult. I don't see there is any good evidence. However, there is evidence that the establishment and dissemination steps, indeed, they are favored by this. And there are several lines of evidence that I don't have the time to summarize, but they are in that review paper I mentioned to you. So that's, the problem of sampling, the problem of some indetermination about what we are doing. So in virus evolution, we have two big areas of research, or two big groups of events that we have to be aware of. The first is the one I have just summarized, quasispecies. That has a lot to do with disease. I'll show you that in a minute. Okay, so this is the result of genetic variation within infected individuals, influenced by host factors, viral dynamics, and so forth. A virus is always conditioned by the host, okay? And that's what we are looking at in our laboratory, and it relates to disease and how to combat disease. But then, of course, a very important consequence of this is when a virus becomes epidemic or pandemic. It spreads in the population. Here in this case, for example, in the cartoon, spreads in the human population. And that has a different approach. Is not to look at mutant spectra quasispecies, but to look at phylogenetic trees, what is the origin of one virus relative to the other, what we call long-term inter-host events, and that's studied by phylogenetic. If you want, phylogenetic is a way to look at phylogenetic trees in movement, okay? And normally, this is done with consensus sequences, not with mutant spectra. Okay? And it's influenced by new parameters, for example, transmissibility, and what we call epidemiological fitness, that we don't know exactly what we mean, but what we try to convey with the term is the capacity of a virus to become dominant. For example, Omicron in SARS-CoV-2 has acquired larger epidemiological fitness than the preceding clades, okay? It's an example. And there are many others, okay? Foot and mouth disease virus, for example, a virus we have worked for many years, has the same. All of a sudden, you have one virus that has high epidemiological fitness, and it displaces the previous ones. Now, a first evidence, or first, if you want, taste of why it is important medically, what I am telling you, is represented in that cartoon. And the essential point of this slide is that the behavior of a viral quasispecies, represented on the left as usual, may be different from the behavior of individual components that you have within that quasispecies. In other words, some cases are very clear to all of you. For example, a virus that is apparently sensitive to an antibody or to an antiviral agent, it may have inside, in fact, it has inside, mutants that are resistant to the antibody or to the antiviral agent, to the inhibitors, CTLs. But there are many other examples in the literature. There are cyclic adaptations to different cell types, a concept that we established in our laboratory that we call molecular memory, that in mutant spectrum remembers what was important for it in generations before, okay? We have altered cell tropism or host range that we would argue in favor of the importance in emerging infections. We have examples in which some individuals can switch into a persistent infection more easily. We have other examples, for example, in vesicular stomatitis virus, natural populations, that you have a virus that is sensitive to interferon, but individuals that are resistant to interferon, and vice versa, that can induce interferon when the whole quasispecies cannot induce interferon, and so forth and so on. So this gives you already a hint that it is important to look at mutant spectra because they contain behaviors that are not the typical that you expect when you just examine the whole population as a whole, okay? So how do we see this in the laboratory? How we really start looking and quantifying what happens in mutant spectra? Well, these are studies that we have done in our laboratory. This is an important work by the group of Dr. Celia Perales here in Madrid. She has her own independent group. In fact, she has made an extremely important connection in our consortium between what happens in disease, in this case, COVID-19 patients, and what we have seen for many years in terms, let's say, of more basic phenomena on quasispecies. Okay, so that link between medicine and, if you want, some more theoretical virology of quasispecies, that's the merit of the group of Celia Perales. So they have started looking. We have thousands of samples that we would like to look for different reasons. Unfortunately, our resources, our money, our everything does not allow us to do all what we like. But an example of what we have done is comparing, for example, patients that have different disease outcomes, okay? Some patients with very severe COVID, others that have mild COVID, others that have moderate COVID, others that have been infected after being vaccinated, and we keep, doing our best in collecting data. What type of data? Well, an example is shown here. This is a fragment of the S gene the surface protein gene of SARS coronavirus 2 of an infected patient from the first wave of disease in Madrid. And what you can see here is that if you put a reference sequence, whatever, we have different reference sequences that we use depending on the experiment. But what you see, and you can see in the slide, all of a sudden, it looks like there are no mutations, but soon or occasionally you find a T or a G or a C, as you can see in the slide. And what we determine is the frequency in the mutant spectrum with which the haplotype in which that particular mutation appears is. And you know, in viral quasispecies, the different genomes, here you can see in the form of mutations or deletions or haplotypes, they reach a frequency that depends on its fitness. And that's a property of the mutant spectra I am telling you about. There is a ranking of mutations according to fitness. So this is the type of analysis and of a study that we do. And thanks to a lot of controls, a lot of repetition of different, pipelines, bioinformatic pipelines that I'm not an expert, but we collaborate with an expert, we have achieved now a resolution of 0.1% frequency cutoff, which is pretty good. It allows us to penetrate into the mutant spectrum. But then you wonder, why are we sure that we can reach such low 0.1% frequency level in our studies? How do we know that these are not technical errors, mistakes in the mutations, rather than true mutations that we find in our mutant spectra? And I want to tell you that we are doing as many controls as we think. And I will mention some, but that's a permanent, let's say, an insatisfaction in our laboratory. But let me tell you, clean read coverage, which is around 1,110,000 with that range, and a high quality score given from Illumina, allows in principle us to reach that resolution level. But in addition, in a cohort of 30 patients, 96 out of 97 mutations and all of the deletions were detected with the 0.1% and also with the 0.5% frequency cutoff, which nobody would argue that that's a reasonable, that's feasible, 0.5%. Now, both resolution levels yielded the same distribution of mutations among the three codon positions, similar representation of amino acid subpositions amino acids in data banks, and we have obtained similar results in duplicate sequencing run, not identical, but very similar, okay? Furthermore, the dominant mutation types that we find at low frequency, those that would be in danger of being mistakes, are T, C, and AG mainly, which are infrequent as the errors that technically Illumina tells us that they produce, which is a change between A and C or changes between CT. There is a reference. And if you are not completely convinced, I suggest that you read the 2 papers that we have published this year, where these controls and additional ones are detailed. Now, I must admit that even consulting with bioinformaticians and consulting with Illumina, I am not able to tell you if I am sure that the number of correct mutations is 90% or 80%, or 70%. I have to admit, I cannot tell you that, okay? And we have consulted with people even in Illumina. What I can tell you is that the majority of mutations are correct, the majority, whatever it means, majority. Otherwise, all these things that are on the slides would, and in the references, would not happen. So when you look at the mutant spectra of SARS coronavirus 2, is really impressive. And you have here, for example, 4 amplicons of NSP12, which is the polymerase, an enzyme that we are interested for obvious reasons is the enzyme that replicates the RNA, or the surface protein, which is on the right. We have the surface protein because, of course, it has to do with immune response, is the component of the vaccines, and so forth and so on. And you cannot see that detail probably, but you can look at the publications if you are interested. These yellow squares mean all the positions where a low frequency mutation or deletion is present. And that low frequency is in the range between 0.1 and 0.49. And the others, different colors that are here, blue and something, and etc., means higher frequency mutations that we find. So you see 38% of the mutations that we examine, of the positions to examine, are mutated, and 97% of mutations are represented in the consensus sequences of data banks, which again reassures us that probably these mutations really exist in reality and are correct, except that are present here in the mutant spectrum. With this mutant spectra, we are doing several things, and with collaboration of Soledad Delgado and Federico Moran from Universidad Politécnica of Madrid, we are applying now neural self-organized maps to see the distribution on a two-dimensional square that you see on the left, and a third dimension, which is the frequency, the distribution of all these different haplotypes that we have in the mutant spectra. So each one of the plots that you see is for one amplicon distribution of haplotypes and one single patient. Okay, and we have seen different sum classes, and each sum, so you see in one amplicon of one individual, each sum is unique. It has a mutant spectrum which is unique. And what I am going to tell you next, that this has functional implications, we have done not with mutations that occur only once, just in case indeed that particular mutation was an artifact. So we have looked at minority mutations, but minority mutations that have occurred several times in different patients, which renders it extremely unlikely that this is the result of a sequencing mistake. And with our colleagues in Barcelona, Núria Verdaguer and Cristina Ferrer-Horta, they have helped us in the following: to locate the amino acid substitutions that we find in mutant spectra in this three-dimensional structure of the replication complex of SARS-CoV-2, which is the minimum replication enzyme. It's called the holoenzyme, that is composed of three subunits: NSP12, which is the one that has the catalytic site, NSP8, and NSP7. And when Núria and Cristina look at the mutations, they say, aha, this mutation may be important because it is connected with a catalytic site in that way or the other. And another thing that they have discovered that is described in this second paper, the PNAS paper that we have just published, is that there is a very important interface between NSP12 and NSP8, where the two subunits touch each other, that is extremely sensitive to modified RNA replication. Okay, so in this type of approach, what they have done is they have chosen the important amino acids according to the anticipation of effects due to their location in the structure, and constructed the gene, express the mutated polymerase, and look at the activity in an RNA primer extension assay. And this is what you see on the left, with different mutants, with a control, at 2 different temperatures. They use usually in this type of experiments 37 and 32 degrees, probably because different locations of the virus in the organism may favor one temperature or the other. And you can see, and the conclusion is that indeed these mutations that we find several times incipiently in patients that have not yet been dominant in the epidemiology of the virus, nevertheless, they are relevant. In the cases that I am showing you, they decrease RNA synthesis. That, as you know, a decrease in RNA synthesis, for example, would be a possible predictor of a tendency of the virus to produce persistent infections and so forth. And although we have not done experimentally because we don't have the tools to do that, but from looking at the literature, we also find that there are predictions of functional relevance for substitutions in the spike protein, as it says here. So I'm changing now topics a little bit. We are still with mutant spectra and quasispecies and SARS coronavirus 2. But there is something that is starting to be evident here now. And the first that discovered this were not us, was a group from China, San et al. This the first reference that I have here. And the idea is the following. In the mutant spectrum of viruses from one clade, you find residues that are not typical of that clade, but they are typical of clades that will appear epidemiologically only years later, 1 year, 2 years later. In this slide, we have such what we call clade discordant residues in mutant spectra with their frequency. That's the top panel with the colors. And below, we have drawn the residues that serve to define different clades of the virus. For example, I don't know if you can see, you see alpha, beta, gamma, delta, 221I, and so forth and so on, the different clades. And you see these black dots are mutations that the WHO, World Health Organization, has served to define the clade. So look how interesting. We find in clades that have nothing to do, that in the consensus sequence, you have no idea that they exist, but in the mutant spectrum, we have combinations of mutations and deletions that are typical of a virus that will come 1 or 2 years later, whatever. And here, for example, in that slide, you can see in Martinez, Gonzalez, Brenda et al., by the way, a student who will present her doctoral thesis today from 2 days from today, on all these subjects. We go from clade 20 to clades 21, 22, 23, and it's fantastic because it may mean several things. One of the things that we thought at the beginning it may mean is that when really people do an isolation and do a diagnosis, perhaps that isolate is also a mixture with some cryptic lineages that are at low frequency and that they go unnoticed. Well, you see, the next experiment that we have done, that's still unpublished, we are preparing this manuscript now, we have taken from the first person who was infected in the US with SARS coronavirus 2 that isolate from 2020, is called the USA WA1, and we received a preparation from BA Resources. We prepared our stock, and then look how interesting. We subjected that stock to 3 plaque-to-plaque isolations, and between each isolation, we treated with mild detergent to disaggregate possible clumps of particles, possible aggregates. And look, in that 3rd plaque that I am drawing here, and we have done it with more plaques, okay? It's not only one. Then we have the same thing. that mutant spectrum of the plaque has sequences that are typical of something that only came later. WA1 2020 is from January 2020, and we find discordances with alpha, beta, and you see the dates. Lambda, December 2020, gamma, January. Fantastic, fantastic. So we think that it is not possible that we have these cryptic lineages to explain this, because we would never have those cryptic lineages maintained in 3 successive plaque isolations. This is essentially impossible. It's one of the few things in science that I would dare to say that it is impossible, okay? You cannot have cryptic lineages that are from lambda at a time where the person who was infected in the United States, these lineages were not circulating, and after 3 plaque isolation. So we come to the conclusions, 2 interesting conclusions that we are working on, so that will be the only thing I will say. First, that probably this reflects a limitation of the SARS-CoV-2 genome to explore what we call sequence space. So it's a huge virus. Mutations are deleterious to such a huge virus. And so it cannot explore, it cannot have any sequences that it can produce the polymerase. So there is a kind of limitation of the number of sequences that are compatible with a viable genome. And thus, if this is true, then we have a fantastic possibility, which is that the composition of mutant spectra may perhaps predict which will be the most frequent or likely sequences in future stages of the pandemic. And that's one of our hypotheses. I don't know, of course, if we are right or not. It could be yes, no. The situation could be still too complex to make prediction. But I think there is some suggestion that may be a way, that would be fantastic, because it would mean that mutant spectra, in addition to the medical implication, it will also have some predictive value, to anticipate possible sequences, possible vaccine compositions, and so forth and so on. So, to go towards the last part of my talk, recapitulation and implications until now. Nasopharyngeal SARS-CoV-2 isolates contain complex mutant spectra with predominance of low-frequency mutations and deletions. Incipient diversification is functionally irrelevant. Mutant spectra include point mutations and deletions that are typical of the consensus sequence of future lineages. Clade discordant residues are found in plaque isolates of WA1 2020. And these observations suggest the control of SARS-CoV-2 poses similar challenges that the control of other RNA viruses. It is not true that because it's a coronavirus, the variation is limited. It may be a bit more limited, but certainly there is lots of variation. Okay? SARS-CoV-2 may replicate very close to the error threshold for maintenance of its infectivity and thus be susceptible to lethal mutagenesis. And third, mutant spectrum may predict sequences of futures. These 3 points are under investigation by our consortium. So let me refer to go into the last 10 minutes or so of my talk. What are we doing? I mean, it's obvious that this variation, mutant spectra, quasispecies dynamics, capacity to adapt, and so forth, is a problem for disease control. And of course, the way to try to combat this has been several combination therapies, as you well know, HAR for AIDS, DAA for hepatitis C, splitting into induction and maintenance regimens, targeting of cellular functions. There are some drugs that are being explored because in addition to being antiviral, they stimulate immune response. People have suggested to combine immunotherapy and chemotherapy. In fact, sometimes this is done, antibodies or monoclonal antibodies with drugs, whatever. This may help, but it is not really a solution. We always have the problem that a control of a disease caused by a variable RNA viruses escapes us. There are lots of escapes. It's not completely satisfactory, despite of the successes with hepatitis C, with HIV, and so forth. So, because we think that quasi-species is important for the understanding of viruses, we have also used quasi-species to apply a new strategy that consists of extinguishing viruses because of the excess of mutations. And that's called transition of the viruses or entry of the viruses into error catastrophe, that is crossing an extinction threshold akin to the error threshold of quasi-species theory. So to give you a rapid view of this, I'm not going to use any mathematics, but there is a relatively simple mathematics that explains this. But you see in that drawing, what it means is that if you have a reasonable fidelity on the left, you have the quasi-species distribution. If you decrease the fidelity of replication of the virus, you go through different steps, lethal defection, overt lethality, until you cross an error threshold, which is predicted by quasi-species theory, in which the virus disappears, because the sequences that make sense, they cease to make sense. They no longer can complete the replication cycle. You can call it no information, random sequences, and so forth and so on. So the error threshold can be viewed as the maximum mutation rate at which the dominant or master sequence, I have not talked about master sequences, but imagine it is the most fit in the distribution and the ones that keep a certain order in the distribution, okay? And they are considered to stabilize the mutant ensemble. When that superiority is lost, then you go into the error threshold. So lethal mutagenesis is virus extinction by an excess of mutations. And what is the basis? Well, the basis is that these drugs, many of them, in fact, all of the ones that we are using in the laboratory are licensed for human use. So it's not only a game of laboratory. They use by mispairing. You have, for example, the case of ribavirin. There is a single bond in this in the structure of the base of the ribavirin. Ribavirin is a nucleoside, but that's the base. And you see that that movement can make ribavirin to pair with either U or C, so it becomes mutagenic, and that's the basis of all of this. There are several now that are being tested, in fact, several, and we have counted up to 19 different viruses that in assays, in vivo or in cell culture, they really are susceptible to lethal mutagenesis. And in that scenario of lethal mutagenesis, we are interested in developing what we call synergistic lethal mutagenesis, that you can easily imagine what we mean. So we are interested because lethal mutagenesis is a broad spectrum antiviral strategy. It has proven effective with at least 15 different viruses in cell culture in vivo. I just said that. Synergy between two drugs means that the combination is more inhibitory than the equivalent dose, but administered with only one of the drugs. The alternative outcomes are additive or antagonism. And several studies have documented that synergies, of course, can be applied to SARS-CoV-2. People have done lots of work before we did ours, and this is an example, that paper of 2022, where there are several discussion of several synergistic combinations. But we had a precedent in our laboratory, which is interesting, is that we showed synergistic lethal mutagenesis with hepatitis C virus some years ago. This is the paper, 2019. And in hepatitis C, we produce virus of high fitness. And we also discovered at the time that high fitness was a factor of resistance to drugs. So it's not only a virus becomes resistant, not only when it has a mutation that gives resistance to a drug, but also when it has high fitness, okay? So we said, let's test favipiravir and ribavirin with low and high fitness hepatitis C, and you see from this slide, And we use two computer methods now for to test synergy. One is called the CompuSyn method. I mean, it's a computer system of Chu, who was the author who developed that. But you see how interesting, if you look at the yellow line on the panels of the top left, the yellow line means the use of the combination of favipiravir and ribavirin. And that combination extinguishes the high fitness hepatitis C. The HCV P200 is the virus that has high fitness, while the two individual drugs do not extinguish this. So with that precedent, we then studied SARS-CoV 2. We measured, we studied whether ribavirin was mutagenic. It was. Here is the typical IC50, CC50. We saw that indeed with our panels of mutagenesis, the ribavirin was really mutagenizing, increasing mutation number and so forth. That's the work of the group with Pilar Somovilla, with the paper that was published last year. And interestingly, there is a synergistic effect between remdesivir, the drug which is most frequently used in treatment of COVID-19, and ribavirin. And this has been done in several ways. You see, if those of you familiar with pharmacology will recognize that this therapeutic index 26 or higher than 10 are good numbers, okay, means that this is effective. And we have two different ways to quantify the synergy, by CompuSyn or the ZIP synergy score that has been published recently. And we are extremely encouraged now, first, to prepare, for example, SARS-CoV 2 of high fitness, and then test new lethal mutagens to see if we can come with synergistic combination. Of course, we have our work restricted to cell culture. We would like very much to go into animal experiments. We don't have the possibility, we don't have the funding, essentially. So if somebody hears me and likes to, engage in some collaboration with animal work, will be fantastic. We cannot do that. But we have the idea that indeed we really have the prospect that synergistic lethal mutagenesis may be good for that. We have some strict rules to declare a virus extinct. And this is, I'm not going to go into details, but we have blind passages, we lose infectivity, we lose RNA. So everything is below at least the detection level. And this is, again, to show that we do positive and negative controls for PCR, highly sensitive PCR. And as a summary of that part, and with that, I am going to the end of my talk, how we distinguish lethal mutagenesis, because there is an increase in population complexity, because there is some mutational bias depending on the mutagenic agent and the virus. There is a decrease of specific infectivity, the ratio between infectivity and the amount of RNA. The consensus sequence remains invariant because there is no selection, there is only a collapse. The error threshold depends on the size of the viral genomes and replication mechanism. And this is actually a very active field of research in theoretical and experimental research. And I have put here 3 articles where you can find more about it. So the conclusions of my talk are the following. Quasispecies dynamics applies to SARS-CoV 2 despite being a coronavirus that encodes an exo-N activity. SARS-CoV 2 mutant spectra are populated mainly by low-frequency haplotypes. There is a deficit of the intermediate frequency mutations as compared with the hepatitis C population, suggesting an average negative effect of mutations and deletions of viral fitness. The virus can be extinguished by combinations of remdesivir and ribavirin in cell culture. The results open the way to investigation of synergistic combinations among lethal mutagens or among the latter and other inhibitors for COVID-19 control. And we have very important precedents, HAART for HIV infections and DAA-based therapies for chronic hepatitis C, which offer encouraging precedents. And as I promised at the beginning, why to be or not to be? Okay, so each replicating genome has a fleeting existence because mutations happen so frequently that you don't have a true lineage in the phylogenetic sense. You don't have a phylogenetic line. But the moment you have a genome with a sequence, the next round of replication, you have a high probability of having another mutation. So that sequence actually does not exist anymore. So a specific sequence, which at a given time is, soon ceases to exist. All is inevitably becomes is not in viruses. Furthermore, the large coronavirus genome has a limit tolerance to mutations. So lethal mutagenesis is expected to push the complex SARS-CoV-2 collectivities from to be to not to be. So second justification of my title, okay? And of course, one has to be cautious. Time will tell if these approaches are of some use for combating emerging viral disease, which is one of our hopes. I think to look at viruses with that magnifying glass of quasispecies, I think it is extremely helpful. But how helpful, time will say. Of course, I give a talk, but many people are behind this. Celia Perales, I mentioned already, critical to have made the connection between medicine and quasispecies. Ana Isabel de Ávila lives essentially inside the P3 laboratory. Maria Eugenia Soria is essential for dealing with the data, management of the data, and so forth. Several students have contributed a lot. Carlos Garcia Crespo, Brenda Martinez Gonzalez, who is presenting her thesis, as I said, 2 days from now, Pilar Somovilla. Then we have the people on the neural network plots that I mentioned, Soledad Delgado, Federico Moran, Ramón Lorenzo Redondo from Northwestern University, extremely helpful, Cecilio Pegalidez, Claudia Aguilar. We have also collaboration with people more on the theoretical part of viral dynamics, Josep Sardañé, Santiago Elena. I mentioned Nuria Verdaguer, Cristina Ferrer-Ortiz, Sergi Vázquez, as people helping, with the structure, and lots of help by bioinformaticians. Carmen Ayuso, Ignacio Gadea, these are people in the medical side, also with, with medical and bioinformatic implication. And a lot of help from our colleague and good friend, Luis Juanes, who is the big expert on coronaviruses in the world, but also in Madrid. He is neighbor to us. And of course, we did not know anything about coronaviruses 4 or 5 years ago. And thanks to Luis and his group, we know now a little more. So thank you very much. And I'll be happy to respond to questions if I can, if I can locate them or see them, because at the moment I only see my screen. Thank you.
No, thank you, Dr. Domingo, for a fantastic and enlightening talk. I will help by addressing questions. Anyone listening, please feel free to put them in the Q&A box, and then I'll address them to Dr. Domingo. So Flor asks that you mentioned favipiravir, but not molnupiravir. Was there a reason as to why favipiravir? Those are hard to say.
No, it's a good question. We have some data on molnupiravir, except that we cannot yet, consider it definitive. And molnupiravir has been used by people. It's mutagenic, and it's probably one that we should consider in our synergy work. So the question is relevant, but I did not show because what we have done is not yet solid enough to really give you some insight of how it is behaving and how it would behave in synergy. So good question, but we don't know yet. But it's truly an important drug or an important inhibitor for COVID-19 treatment. Yes.
Thank you so much. Anna asks, are there well-identified specific mutations related with the organ in which the virus is replicating? So
Not that I know of. Not that I know of. This is an extremely difficult issue, very important, because, when a virus replicates in an organ and not in another, it has at least 2 components. A chance component that which virus entered the organ. Okay, you imagine you have a situation of viremia, have lots of virus circulating, and then first of all, a certain phenotype is usually not exclusive of one sequence. In other words, probably I have several alternatives for a virus to increase fitness, several alternatives for a virus to feel well, for example, inside the lung rather than the upper respiratory tract. So I think it goes against what we know about virology, that we will be able to say that sequence is lung, that sequence is bronchi, or that sequence is in the brain because it's neurological disease. Of course, there are some examples in virology which suggest that something might happen. As you know, in measles virus, subacute sclerosing panencephalitis is a complications of measles that goes into the brain. And then if you look at the sequences, they are all hypermutated with certain genes. And that's so you can argue the same way that you have measles in the brain in subacute sclerosing panencephalitis you could have. But I don't think that will be that simple. So again, a good question, but I am not optimistic that we can catalog sequences according to where the virus replicates in the organism.
I think that's fascinating. Now, Julian asks, first says, awesome talk. And how far are we able to use quasi-species as a predictive measure for evaluating emergence potential of some viruses?
This is an extremely relevant question, very difficult. And I think what we have to start doing is to put these sequences that we think that they may have a predictive value, okay, put them in the algorithms for prediction that I don't know, I must confess, I don't know anything about, but I know that they are interesting, and I would like, to have collaborations. We hope to have soon a collaboration in this respect. And if we, first step, are we able to predict something with the tools that we have now? Then second question, the world of coronaviruses, because we are now looking only at a small part of the coronavirus, which is SARS coronavirus 2. But we have sequences of other human coronaviruses, SARS, MERS, other, different coronaviruses thing. Then the question is, how do all of these differences have something in common? Do the sequences that we have in our mutant spectra are also present in other coronaviruses? And then once we know a little more about that, I have in my mind something like 5 to 10 years from now, then we may dare to see if we can predict some emergence. But again, extremely difficult. I think a lot of prior work is necessary before we can see the reliability of these approaches and go to say, look, the next coronavirus is likely to have, a spike like this and a polymerase like that. I would like to be able to reach the point where I can do that, but I'm afraid I won't see it.
No, I think that's a really helpful perspective. And Alan asks, could host range restriction be considered a variety of the extinction threshold, rooted in the number of host factors that restrict a particular virus?
Yes. I mean, an extinction can come about by an excess of mutations, but also an extinction can come about by limitations that the virus has in its usual spread to go on, replicating. So if all of a sudden a virus finds itself with a group of cells that do not expose a receptor or a co-receptor or whatever factor inside the cell that is compatible with its entering the cell or its replication, of course, that will be a factor of extinction. In fact, bottlenecks can be viewed as a factor of extinction. If you subject a virus to different bottlenecks and you decrease fitness through bottlenecks, again, that virus will be more susceptible to extinction by the things that we do, this error catastrophe I told you and so on. So the question is, yes, if we have any kind of barrier in the virus, it helps to be extinguished. For example, you have put the example of the receptor restriction, but another way of looking at the same problem is if I do inhibit the virus, if I lower the viral load, that virus will be also more susceptible to extinction.
Absolutely fascinating. I think we're right at the hour. I want to thank you, Dr. Domingo, for such an engaging and intriguing presentation on quasispecies. Thank everybody who participated in the question and answer session. That discussion really added a lot to our insights today. And I look forward to seeing you all at the next Forefront of Virology webinar series. And thanks again so much to Dr. Domingo.
Thank you for the invitation. Thank you all.